So now we're going to get into meiosis. It's very similar to mitosis, not just in how it's said. Okay, science didn't really help out too much with keeping them separate because mitosis, meiosis, yeah, a little confusing. But meiosis basically is going to make 4 different cells. Okay, instead of 2 genetically identical cells, we're now going to go through and make 4 genetically different cells. So, like I said in the previous video, meiosis is actually split into 2 separate processes. Okay, so you've got meiosis 1 and meiosis 2. So super, inventive naming system here. Meiosis 1 is set up to divide the homologous chromosomes. Okay, so it'll basically take it so that you start off with 2 copies of each chromosome, but by the end you're going to have 2 cells that each have 1 copy of each chromosome. So you're going from a total number of 46 chromosomes down to 23. Okay, and it is split into, again, prophase, metaphase, anaphase, and telophase. Only now, because it's in meiosis 1, it's prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, so this is a pretty good summary of the whole process. Feel free to come back to this slide, but we're going to go through each individual process, each individual step in the process in a little bit more detail here. All right, so prophase 1. Again, this is really similar to what you saw in prophase in just mitosis. So first up, you've got your nuclear envelope is going to break down. So this is so that the chromosomes can actually get out of the nucleus into the cell where they can be kind of messed around with. All right. So this is showing that you have 1 copy of each chromosome from each parent. Okay, so it's showing the purple and the green chromosomes are the homologous chromosomes, with the purple coming from the mother, the green from the father, and then the teal and the red are also homologous chromosomes, with the red from the mother and the teal from the father. Okay, this is, again, very simplistic because it's just showing a max of 4 chromosomes instead of all 46. But you can see here that our nuclear envelope is beginning to break down. So that's going to release the chromosomes out into the middle of the cell. You also have your centrosomes beginning to move, okay, towards opposite ends of the cell, and you're going to get your spindle fibers to form. Okay, because remember, the spindle fibers are really important for division because they will actually attach to the centromeres on the chromosomes to pull them apart. And then you're, of course, also going to see the duplicated chromosomes condense. Okay, so these are duplicated chromosomes. Okay, so you do have your sister chromatids, right, which is why you have this X shape. Okay, because remember, anytime you have an X chromosome, okay, X-shaped chromosome, not a chromosome X, but an X-shaped chromosome, it is duplicated. Okay, so you do have your 2 sister chromatids. So this is actually when you're going to first see the homologous chromosomes start to pair up. Okay, so chromosome 1, both copies of chromosome 1 are going to start pairing up. Both copies of chromosome 2 are going to start pairing up, so on and so forth, all the way down the line. Okay, now this starts really early so that by the time it gets to the point where they're going to actually separate, they pretty much have had time to line up every single homologous chromosome. Okay, so next up we go into metaphase. So during metaphase 1, you're going to have the spindle fibers aligning the homologous chromosomes in the center of the cell. Okay, so you can see, obviously, we have the center of the cell marked by these dash marks, and we have 1 copy of each homologous chromosome on either side of that center mark. Okay, what you'll notice, it doesn't matter what side of the cell it puts the chromosome that originally came from the dad or from the mom. Okay, because we said that the purple and the red were from the mom, and the green and the teal were from the dad. Okay, at this point, it really doesn't care the distinction of where these chromosomes came from. Okay, just so long as they have homologous pairs and they pair up during this stage. Okay, so each side of the equator has chromosomes from the organism's parents. Okay, so from both of them. All right. This is the final stage where the cell is both diploid and has 2 copies of all the DNA. Right. So now we're going to go into anaphase 1. So in anaphase 1, you get the paired homologous chromosomes separating from each other. Okay, so the 2 chromosome 1s are separating from each other, 2 chromosome 2s are separating from each other, all the way down. Okay, so now you're going to end up with 23 chromosomes on each side of the cell. And you're going to get a split right down the middle between them. Okay, so the homologous chromosomes move towards opposite sides of the cell. Important thing to remember is that each of these chromosomes is still comprised of 2 sister chromatids. So it is still duplicated DNA. Okay, so the sister chromatids remain attached to each other at the centromere. So the cell starts to become haploid with 2 copies of DNA on each side during this stage. At this point, we're going to go into telophase 1, which kind of also contains cytokinesis. And at this point, we're going to have the spindle fibers start to disassemble, okay, because they have gone through that first round of division. So they're kind of happy with what they've done now. You're going to get the cell membranes start to reform. Okay, the cell undergoes cytokinesis. And you're also going to see that you have 2 separate cells. Each cell is haploid, okay, because each contains half of the number of the total chromosomes, okay? But each of these chromosomes still contains 2 sister chromatids, so they're still duplicated chromosomes. After telophase 1, the cells are actually going to enter a period of rest, so it's very similar to interphase, okay? Not quite as intense, not quite as long, but it is a period of rest before they continue into meiosis 2. So, summary of meiosis 2 is very similar, except now we're going to be showing 2 cells going through it at the same time. Okay? So we start off again with prophase 2. So this is where your nuclear envelope is going to dissolve again. Okay, so it's been having kind of a rough time, keeps reforming and then getting broken down again. Okay, so our nuclear envelope is dissolving and our spindle fibers are reforming. Okay, centrosomes are moving to opposite sides of the cell and getting ready to undergo another step of division, another process of division. Metaphase 2, now you've got the spindle fibers are aligning the chromosomes along the equator. They no longer have homologous pairs inside the cell, so the spindle fibers are attaching to either side of the centromeres. Okay, so they're getting ready to pull these X's apart. So at this point, each cell has only 23 chromosomes. They are haploid. They are still doubled, okay, duplicated, so they have the sister chromatids. Now, during anaphase 2, the sister chromatids are finally going to be pulled apart. Okay, so this is very similar going back to anaphase, regular anaphase in mitosis. You get the sister chromatids pulled apart. Okay, so just remember, anaphase 1, you get the chromosomes, the entire homologous chromosomes separated. Anaphase 2, you get the sister chromatids of those chromosomes pulled apart. Okay, so the chromatids are going to move to opposite sides of the cell, okay, just like normal that we see in anaphase. And this is the phase where the haploid cells separate the 2 copies of DNA. So now you already went through meiosis 1 to get down to haploid cells, and now you're separating the 2 copies of DNA. Okay, so you're getting back to kind of operational mode, okay, without being diploid. Okay, that's still kind of out there. All right, then you go into telophase 2. So again, you've got your nuclear envelope reforming around the chromosomes. The spindle fibers are going to break apart. They have gone through 2 rounds of division in order to get to this stage. They are completely happy and ready for a good rest. The cells will undergo cytokinesis yet again. This completes meiosis. So now, if you look at it, you have 4 cells, okay, 4 daughter cells produced. You have half the number of chromosomes that you started off with in each, and each only has 1 copy of each chromosome. Okay, now these at the moment look like these 2 cells are genetically identical and these 2 cells are genetically identical. We'll go into crossing over later. That explains why they are not actually identical. Okay, so there is a process called crossing over that takes place, but we'll get into that later. So it takes 1 diploid cell and goes through 2 rounds of division to get to 4 genetically diverse haploid cells. Okay, and that is the main purpose of meiosis. So this is the summary of the entire thing. We have meiosis 1 dividing the homologous chromosomes, separating them from each other. And then you go into meiosis 2, and you take those 2 cells that were produced in meiosis 1, divide them again, and now you're down to haploid cells that only have a single copy of the DNA. Okay, so meiosis differs from mitosis in several significant ways, and we've kind of touched on these a little bit. So first up, meiosis obviously has 2 rounds of cell division, while mitosis only has 1. Okay. In mitosis, the homologous chromosomes never pair up. They really don't care where they are in relation to each other in the cell when they start lining up for metaphase and then separating out the sister chromatids during anaphase. Okay, so in mitosis, they will divide independently of each other. They're each going to separate their sister chromatids, sending 1 to each side of the cell, but they don't really care what their homologous chromosome is doing. Okay, they assume that it's dividing properly as well, but it's its own business. Meiosis is also going to result in haploid cells, so half the number of total chromosomes. Mitosis is designed to keep the number of chromosomes constant. Okay, so it's going to be diploid cells. So